Okay, I hope everybody is listening well. So my name is Priscila Figueiredo, and I'm a postdoc at Dr. Didier Ronan's lab in the Structural Chemogenomics group at the Faculty of Pharmacy in the University of Strasbourg, France. And I've been here for 12 months in an international comparison program from the University of Rio de Janeiro, Brazil. So in the first moment, we'll just talk briefly about our structure selection and the protein ligands preparation. After, we talk a little bit about our docking and rescoring protocol for this challenge, and how we performed, talking about the post-prediction accuracy and also our failures. And finally, what we have learned from the challenge and also the advantages of the method we used. So the system setup consisted of selecting the available structures from the protein databank that were ligand bound, so 26 in total. We added the hydrogens for these complexes using Protos after we verified the protonation of the active sites manually. Also, we extracted the ligands that were coxtralized and we eliminated the loosely bound water molecules. And for the preparation of the FXR ligands, we generated 3D coordinates using Carina, and we assigned the protonation using filter, and always verifying manually for possible mistakes. After the preparation of the molecules, we used the Soflex dock to rigid docking these FXR ligands to each one of the 26 structures we selected. Soflex dock, it's a rigid docking approach by incremental construction of the ligands inside the binding pockets, and it uses protomol, that it's a pseudo-target molecule to dock the ligands that can be, for instance, residue-based. So basically, we aligned all these structures together, and we kept the residues that were located in a sphere of 6.5 angstrom around the coxtralized ligands to use as a residue list to make this protomol. We also use the PDOM option for the docking runs that assures extra accuracy for the docking process by minimizing the ligands inside the binding pocket. And we generated 20 poles per each ligand, which gave overall over than 53,000 poses. After, we are going to use a method developed in our group to rescore these docking poses. So we used a method called GRAIN that is based on the knowledge of protein-ligand interaction patterns that are contained in known crystallographic structures of ligand-bound proteins. So we are going to use this information of known structures to generate an alignment-based interaction with the target complex for which we want to rescore a docking pose. So briefly, the method will rely on interaction pseudoatoms that are placed on the interaction atom on the protein residue, on the interaction atom of the ligand residue, and in the center between them. And if we generate a set of IPAs, interaction pseudoatoms, to the ligand, to the complex that we use as a reference, that is the non-coxtralized complex, and the target complex that we want to rescore the pose, we can make a graph. And this graph will have as vertices the IPAs and as edge all possible links between them. So if you align these graphs using a graph matching algorithm, we are going to obtain a product graph in which each vertex would describe a match between a reference IPA and a target IPA only if they have the same kind of interaction. There is either a hydrogen bond or a hydrophobic interaction, and so on. So the quantification of this alignment will be done through the GRIM score. And the GRIM score is an empirical scoring function that it's based on the number of matched ligand-based IPAs, the number of matched centered IPAs, the number of matched protein-based IPAs, also the sum of pair weights in each click of the graph, divided by all possible pair weights, the root mean square deviation of this matched click, and the absolute value of the difference in the number of IPAs between reference and query. So in practice, if you have a GRIM score, for example, superior to 0.8, it's an indication of a good match between reference and target. That would mean that they have the same binding interaction mode, not necessarily the same binding, the same chemical structure. As you can see in this figure here on the left, where we see that, for example, the hydrophobic interactions of reference and targets are well aligned, represented by cyan and green dots, and also the polar feature of these ligands represented by dark and light blue and dark and light red. So my slide is not skipping. Okay. So in practice, what we did was take all these docking poles that we have, and we pre-filter them by reducing, eliminating the poles that have a Suflex score inferior to 2. And we did pairwise comparisons with GRIM calculation to each of the 26 FXR ligands that were co-crystallized to the receptors that we used. And we chose the 5 past poses based on their values of GRIM score to submit. So in the overall pose prediction, we see that we didn't have a real docking problem, since in the overall sample, that is the sample that was not pre-filtered by the docking score, we had poses that were lower than, that had RMSD lower than 2 angstrom, but we had a real scoring problem. But still, GRIM score is able to outperform slightly better than Soflex score, as you can see by the average RMSD for the top scoring poses in the graph. If you analyze ligand by ligand, we see that we can correctly score the benzimidazole series of the FXR ligands. And this is quite logical because we had 7 of the receptor templates that we used in the GRIM calculation containing benzimidazole. So it makes sense that GRIM would match the binding mode of these molecules. We also noticed something interesting from these comparisons. We saw that there was a strong relation between chemical similarity to the reference ligand and the RMSD. And nevertheless, we didn't have a higher correlation between a high value of GRIM score and a lower RMSD poles compared to the crystal structure. So we see that we have high GRIM score that doesn't correspond to a good prediction, only very, very high GRIM scores corresponding to a good pose prediction. And we've investigated the reasons for it. So one of the outputs of GRIM score when you calculate it's the number of matched polar interactions between the reference and the target. So we see that this number is correlated to a good prediction or not. So we had repeated failures when we had a higher GRIM score but no or few polar interactions to match between reference and target. And since the nature of this protein was very hydrophobic, we couldn't use N-polar as a cutoff to eliminate these poses because we had several poles that had 0 polar interaction in this case. So of course this was one of the reasons for failure in this challenge, where we predicted this pose with a higher GRIM score, but they had no polar feature to match between the reference and the target. Here on the right, it's the illustration for FXR8, where we have a higher GRIM score, we have a higher similarity to the reference that was used in the alignment, but we noticed that there is a flip on the orientation of the ligands inside the binding site, so we missed this polar interaction to actually predict correctly these ligands. Also, we noticed that if we don't have a similar structure to compare, or at least a structure with a similar binding mode, the GRIM will fail. The docking problem was just for 2 ligands where Surflex was simply incapable of finding a good pulse that had at least RMSD inferior to 3 angstrom. And we also noticed what you can call a different binding mode for FXR2. So here on the right, we have in cyan the top predicted pose by green, aligned to the structure that we use in green calculation to give this pose in orange. So we see that we have a higher green score for this match. It would make sense that they bind the same way, making a hydrogen bond with a tyrosine residue in the active site, but in reality, the crystal structure in green binds completely different, making another interactions inside the pockets. We also notice a flip on the side chain of a leucine residue inside the pocket, so induced fit in this case was very important to a correct prediction of these poses. Nevertheless, we were not so bad compared to the other predictions. So if you notice the graph of the RMST for the top scoring poses, we are in 14th place. But now I would like to talk about our ranking for the stage 1 of the challenge, in which we use the height scoring function to rank this ligand. So just going briefly, height is a scoring function based on the energetics of dissolution, and it takes into account the local hydrophobicity, the solvent accessible surface, and the contact surface of the pocket. These hydration and dissolution terms are determined using octanol water partition coefficients of small molecules, so they are not calibrated on affinity data, and in principle, it's applicable to every protein target. So simply what we did, that we took the top predicted pose by green, that is the pose with a higher green score for each ligand. We calculated the high affinity score, and then we ranked these poses according to their high score. So we can see that we are in 3rd place on the general prediction in this challenge. So mainly what have we learned from the challenge is that green helps to rescue bad scored poses. We noticed that for some of the ligands, he was slightly better, but for other, he was much better than Soflex scoring pose. So it really helps to rescue these really badly scored poses. Unfortunately, green will really rely on the similarity of binding mode to the templates. So if we don't have it, we don't have a good prediction. And we also highlighted the importance of polar interactions to have a correct alignment. So not relying just on the green score value. For the structure-based scoring, we saw that post-selection by green and ranking by height could be an efficient strategy, and this protocol is fast enough. Green takes only seconds, such as height, to calculate for each pole, so it could be applied at a high throughput scale. The advantage of using this method is can be coupled to any docking program to post-process poses. It takes advantage of ligands that have the similar binding mode but not necessarily similar chemical structures. Could be, for example, applied in a target family biased strategy. And the program is a model of the iChem toolkit, so if anybody is interested in downloading, this is the link. You can just send an email to Dr. Didier Ronan for asking for a license. And here are a couple of references if somebody is interested in the method, and also how it performed in the last challenge. So I'd like to thank Dr. Didier Ronan for giving me the opportunity to give this talk in his behalf, Guillaume and Frank that helped me somehow in the processing of the poses, and you for your attention. Thank you very much.